Okay. Hello all, this is Rajneesh Khambati. Okay, today we are going to learn chapter the fundamental unit of life from standard 9. Okay, so let's begin. This is PowerPoint presentation made by me. So let me open presenter view now. Okay, now before we start, students, please take your notebook and pen and pencil with you so that you can make some notes, okay, for your use to read. Merely looking at the video won't help you. Okay, so let's begin. You can see this is nice animated picture of the cell. Okay, you will understand everything by the end of this video. So let's begin. We will start with some introduction. Okay, let me put few lines now. See, scientist number 1. Okay, the very first, Robert Hooke, in 1665, he saw the cork. Okay, what he saw? He saw the cork. Now, cork means, what do you mean by cork? Cork means wood, and wood means dead tissue. Okay, wood means dead tissue. So basically, Robert Hooke in 1665 observed dead tissues, the cork, wood, under the microscope, and he could see the structure looking like honeycomb structure. Okay, and honeycomb structure means it is looking like hexagonal structure. Okay, we know that how honeycomb look like. It is looking like this, a hexagonal structure. Okay, so, and to this structure he gave the name, and the name is the cell. To this structure he gave name cell. Okay, to this structure he gave the name cell. Now the question is, why cell? Because cell means, in the Latin language, you can see, okay, you can see in the Latin language, cell means little rooms, rooms that do not have windows and doors. Okay, there is nothing, only wall is there and there is nothing. So that is why the name is given, the cell, to this honeycomb hexagonal structure. And in that honeycomb structure, each and every hexagonal structure is cell. Remember that. Okay, each hexagonal structure is known as cell. Clear? Means each hexagonal is cell. So this is the first thing we should know. Now, later on, another scientist came, that is Leeuwenhoek. Okay, a Leeuwenhoek is a scientist. Now you can see what he did is, Leeuwenhoek in 1674, with the help of improved microscope. Now see this also. Okay, by this time there were present of good microscope. Okay, so he observed pond water. Okay, what he observed? Pond water. And in the pond water, he could see living cells. Okay, remember this. He observed pond water under the microscope, and in the water, he could see that there is something living, small microorganisms are present. So, if you just basically differentiate between the Robert Hooke and Leeuwenhoek, okay, Robert Hooke saw dead cell, and actually it was just a cell wall. Okay, what he saw? Just a cell wall. Okay, not living cell, dead cell, and that too cell wall. But the Leeuwenhoek observed water, and he could observe living cell. So Leeuwenhoek was the first scientist to observe living cells under microscope in 1674. Now later on, another scientist, Robert Brown, in 1831, he observed nucleus. Okay, nucleus. Now, what is the nucleus? You know that, for example, this is the cell. Okay, and in the cell, there is something present in the center of a cell, which is looking something like this. Okay, so he said that this center mass is known as, okay, this center mass is known as the nucleus. Okay, it is the nucleus. Later on, another scientist, Purkinje, in 1839, he coined the term protoplasm. Okay, what he gave? Protoplasm. Now, protoplasm and cytoplasm, there are two terms in a cell. Over here, protoplasm means cytoplasm along with nucleus. Yes. Okay, along with nucleus. Okay, so that is called as protoplasm. So basically, protoplasm includes all the membrane made structure, like which are made of a lipid, protein, okay, carbohydrates, and the membrane structure. So that all comes under protoplasm. And okay, this is the introduction of the chapter. Now all these things are from your textbook only. Okay, you can read your textbook to see these terms present in your textbook. So now let's move ahead. I hope that you are getting all these things. Okay. Now this is 2-mark question for you. For example, the question is, who discovered cell and how? Let me write the question. Who discovered cell? Okay. And how? So what can be the answer? You now, this is the answer. The as it is, same as it is, you can write it. First, Robert Hooke sliced corkwood into the thin sections and observed under microscope. Okay, what he did is he took the wood and he made very thin slices, and then he made slide of it and then observed under microscope. Now, under the microscope, he could see honeycomb looking like structure. You can see this orange image over here. Okay, it was looking like the same honeycomb structure in orange picture. And to this structure, he gave name the cell. Okay, you can see this all structure, which are a hexagonal structure, are known as the cell. Okay. Now, each hexagonal structure in this whole image is called cells. That means there are many cells present into this structure, and it was looking something like this under the microscope. And again, the line is there that each little hexagonal structure is known as cell, and the cell means, in the Latin language, cell means little rooms, rooms without windows and doors. Now, this is an activity I made for you so that you can have visuals that exactly how it is looking. Okay, I did the same activity, and let me play the video for you. Okay, see, I did the same thing. I took the wood, I made very thin slices of the wood, and then I prepared the slice and observed under microscope, and this is the view of under the microscope of that slide. You can see it is looking like hexagonal structure. Okay, this is the video and this is the image. They both are side by side. You can observe, you can pause the video and have a look at it. Okay, yes, you can see it is looking hexagonal, somewhat sometimes pentagonal structure. Okay, but this all structure in this video or in this image is known as cell. If I give you, you can see this blue arrow, okay? I'm giving you this arrow here. Each structure is called the cell, okay? This is what? This is cell. This is cell, okay? Now, let's move ahead. I hope that you are understanding. Okay, this is the video again, so that you can see that exact view. Yes. See it very carefully and try to observe and also learn how to observe under microscope. Okay. Okay. Next point is cell theory. A cell theory was given by three scientists. It is just three sentences, actually two sentences, okay, which are known as cell theory. Let's read this out and understand. The cell theory was proposed by two scientists at first. Scientist number one is here. Just a second. Yes. Scientist number one is this Matthias Jacob Sladen in 1838. Okay, the name of scientist is Matthias Jacob Sladen. He was the German botanist. He was a botanist, and a botanist always, botanist means who studies the plant. Okay, who studies plants are known as botanist. So Sladen was the botanist and he was studying the plants and all the things about plants. And Theodor Schwann in 1839, he was a zoologist. Zoologist means who studies animal cell. Okay, who studies plant cells are botanist. Who studies animal and animal cells and everything about animal is zoologist. So Jacob Sladen, as he was the botanist, means he observed plant under the microscope, and Theodor Schwann observed animal cells under the microscope. Okay, and both saw one common thing. Okay, both actually they gave separate sentence. Okay, the sentences are like Matthias Jacob Sladen, he was a botanist and he observed plant under the microscope, and he said that all the plants are made up of cells and its product. Similarly, somewhere else in the world, Theodor Schwann, he was the zoologist and he observed animal cells under the microscope, and he said that every animal is made up of the cell and its product. And later on, they both together combined the sentences and said that all the living organisms are made up of cells and its product. Okay, so this is the main sentence we are getting from these two scientists. Later on, the theory was further expanded by German physician Rudolf Virchow. This is another third scientist known as Virchow in 1855. Okay, he said that all the cells are arising from pre-existing cells. Omnis cellula, e cellula is the sentence given by him. Okay, it's a main sentence. Omnis cellula, e cellula. That means all the new cells are arising from pre-existing old cells. You can compare, like there are parent cells, and parent cells if present, then only a new daughter cells can be produced. Okay, if there is no parent cell, there is no daughter cell. Okay, and together we are getting now three sentences. So let's read the sentence. First, all organisms are made up of one or more cells. Number two, The cell is the fundamental unit of life. Okay, the fundamental unit of life, or you can say the cell is structural and functional unit of all living things. And the third sentence is the cell arising from other cells through cellular division. Means the new cells are arising from the new cells are, okay, you have to repeat after me. The new cells are arising from pre-existing cells through cell division. Okay, so this is the cell division term. Now we have to see this term in the end of the chapter. What is the meaning of cell division? Okay, basically a cell division means there is one cell, and that one cell will then divide into two daughter cell, and the process is known as cellular division. So this is the three sentences of the cell theory. I hope that you are getting this. Now we have to do one activity according to NCERT textbook. Okay, so let's see the activity. Now activity goes like this. Okay, we have to take onion bulb. Okay, now I will show you directly video so that you can now what to do. See, I'm taking a slide and an onion bulb, watch glass, okay, and stain. This is the bottle. The bottle is of stain. A stain is a liquid which helps you to colorify the objects so that you can observe them under the microscope, because if they are colorless you cannot see them under the microscope. Now take the onion bulb and like in the video you have to take the inner peel of the onion. Okay, you all see this is the pill. Okay, this is the pill, but it is outside pill. You can take that too. Okay, doesn't matter whether it is inside pill or external pill, but we need that pill. So you can see this is inner pill. Yes, the paper, it is looking like a paper plastic. Now with the help of forceps you have to take the pill, small portion of that pill out. Okay, I have got, this is the pill. Okay, now I am putting that pill into watch glass filled by water. Okay, filled with water with the help of brush. Now the pill is ready. Okay, very clearly try to observe. Now I am adding few drops of safranin stain solution. Okay, safranin is the name of this stain which is red in the color. That means our pill will be looking pink or red in the color. Okay, now then after that take the pill on a slide. This is called as preparation of a slide. Okay, this is called as preparation of a slide. So this is how I am preparing the slide. Then put, wait, okay, wait. Yes, now this is cover slip. Okay, this is cover slip, a very small thin glass. So this is how I am preparing the slide. The method is called slide mounting. The method is called slide mounting. After that, I will put the slide under the microscope and now you can see that. Now this is how the cells of the onion looks under microscope. You can see they are looking like a brick. Okay, they are somewhat brick-like structure. See, very clearly seen it is. Okay, now, okay. Now you can see over here, please pay attention at this. You can see there's something round looking in the center of this brick-like structure. Do you see? This is what? This is nucleus of the cell. Okay, this is the nucleus. Now, in the hexagonal structure, it is known as a cell. as well as this brick-like structure is also known as the cell. Let me make outline for you. This is brick-like structure. Okay, it's a brick-like structure, and this structure is also known as the cell. It is also known as the cell. Okay, and this structure, which are circular, round, oval mass in the center of the cell is nucleus. Okay, clear. Now, observe the video very carefully. Okay, see, we have to go through the slide and we will try to observe each and every cell. Okay, so you can see now this whole structure, what is which is in front of us, is a tissue, plant tissue, which is made up of a cell, and each brick looking like structure is the cell, and there are many cells we can see in this structure. Okay, so this is the activity. I hope that you have got this activity. Okay, yes. Now if you can't understand, you can leave a comment in the video so that I know that whether it is good or not for you, whether I have made it good, okay, or not. Now you can see the same thing I have shown in the picture, that what is the nucleus in this. Let me give you visual for it. Okay, you can see, do you see this, okay, this, this, and here, and here, in here. Okay, you can see this is the nucleus. This is the nucleus. Very clear nucleus is here and a cell. Okay, I will making the outline for you. You can see this is how it look. This is a brick looking like structure. It is a cell. And in the center of the cell, always small circular round mass is present called nucleus. Okay, so now up till now we can understand two things right now. Okay, there will be the cell, shape of the cell can be of any type, but in the center there is always a nucleus is present. And one more thing, between the nucleus and this cell wall and the cell membrane, this space, okay, you can see this is the space, that space is known as cytoplasm. Okay, you can see that cytoplasm space is present in each and every cell. Okay, each and every cell has that. See, okay, so this is the three things: cell, its outer boundary, inside it is filled with the liquid cytoplasm, and in the center of the cell there is a nucleus. Okay, so I think enough, it is enough for you, enough analysis. Let's move ahead. This is the video again of the onion cell only. Okay, this is the onion cell under the microscope, 45x zoomed view. You can see, if I make it zoom, okay, you can see very clear nucleus. This is the nucleus, very clear. And this everything is cytoplasm, okay, surrounding the nucleus. A cytoplasm is there. Okay, now let's move ahead. Okay, we don't need to waste time over here. Now, up till now, okay, these many slides we learned. Now, what did we learn and what did we understand? So see, what do we observe? Now, these are the questions, okay, written here. You have to read the question and try to answer the questions by yourself so that you can understand, okay, what to understand from the previous slides and what you have understood. Okay, so the first question is, what do we observe as we look through the lens? Okay, from that video, if you know, if you look under the lens of the microscope, what we can see? The cells. Okay, we can see the cells. There are many cells present. Then can we draw the structure that we are able to see through the microscope? So can you draw the cell from the microscope? Yes. Let me go back. Now you can see. Try to draw this structure. Okay. Try to draw this structure. Now if you are confused how to draw, I will help you. Okay. Just draw like this. Okay. Just draw this boundary. This is the boundary. Okay, and in a center this is nucleus, and then we have to give it the name. The name is cell wall. This is nucleus, and this is cytoplasm. Okay, three things we can observe in this image. So is it easy? Okay, it is easy to draw. So this is how you can draw. Now let's move forward. Okay, the third question says, what does it look like? I just draw it. Okay, so it is looking like a brick. Okay, like structure. What do we observe? We observe cell. Obviously, we are observing cell. Do we see similar structure or the different structure? This is the question. This, okay. And what are these structure? This last two question. Let us go back again. Okay, in this diagram here. Now, do we see the similar structure? Yes. See, all the structures are somewhat similar. Means looking like a brick, somewhat same structure. Or in the previous slide, all looking hexagonal structure. So they all are looking same. Okay, almost same in this one particular tissue. And what is this structure? It is a cell. Okay, this brick-like structure is cell. Okay, so I think these questions are clear now. Okay, so and if you don't understand these questions and their answers, you have, okay, do one thing, go back. Okay, watch the video again. Do not go ahead first. Okay, do not move. Do not see the video. Go back and see the video again. Try to understand. Okay, once you are able to answer these questions, then you have to move ahead. So right now I am moving ahead. Now, you can say this, there is one more image of the onion cell only, but it is outside pill, which is looking pink in the color. Now, these structures, these are the answers over here. Okay, these structures looking, okay, here. What is this? These structures looking similar to each other. Okay, we can see in this diagram, backside, see the diagram. All the structures are looking similar, same. Then together they form a big structure like the onion bulb. Is it correct? Is it correct sentence? That these structures together form a structure, a big structure like onion. Okay. Then hence, they are called as building block of life. Now, this is very great sentence you have to understand. That is the reason we call the cell as building block of the organism. Why building block? Because you can see in this diagram, there is one cell, okay, one single cell. That one single cell, now likewise, many cells are present in the whole structure. And that whole structure is coming from onion. That means the whole onion is made up of cells. So the cell is building block. What if I remove this cell one by one? Okay, there will be no onion bulb. So that is why it is called building block. It is like our wall, okay, your room wall. In the room, the walls are made up of brick. So brick is the basic structure in the wall. Similarly, the cell is basic structure of the organisms, every living organisms over here of the onion bulb. Then we find from this activity that onion bulbs of the different sizes have similar small structure visible under microscope. Now, if you take a small onion bulb and observe the cells under microscope, and if you take a big size onion bulb and observe its cells under the microscope, okay, you can see that big onion or the small onion, the cells size and the structure is same. The structure is same and the size is also same. It is not like if the onion is big, its cell will be bigger in the size, and that is why the onion is big. Okay, it's not the case. The case is big onion have many more number of the cells, and the small onion is having less number of the cells than the big onion. Okay, the cells of the onion peel will all look the same. Means every peel of every onion, every different onion will be looking the same regardless of their size of the onion, big onion or the small onion. Yes, and that is why we can say that the cells are the building block of the onion bulb, or you can say basic building unit of the onion. Let me underline it for you. Okay, what is this? The basic building unit of the onion bulb. It's called cell, and these things are known as the cell. This is called the cell. Okay, now let's move forward. Okay, now this is one more question. Okay, this is one more question. The question is, let me give you the question. The question is, why? What why? Okay, see, why the cell is called structural and functional unit of life. The question is here. Why it is called structural unit of the life and why the cell is called functional unit of life. Now, these are two different terms we have to understand. We call the cell structural unit because it is forming the structure. Okay, why? Because it is forming the structure. Structure how? Let us see. First, it is formed, there is a cell. See this? Cell is there. Now, all the cells together form the tissue. All the tissue together then forms organ. All the organs together then form the organ system. And such many organ system will form the body of organism. That means the body, this body, what you look at is made up of cells. Cells are the basic building block of this body. So that is why the cells are known as structural unit because they are making the structure. Okay, I hope that this one thing is clear. Okay, now let's move to the second thing, that cell is called functional unit. Okay, what is the cell? Also known as functional. Now, why functional? Functional, simple. The meaning, it is doing something. The function, okay, it is doing some functions. Now, what functions a cell can do? A cell can do metabolism, or which is also known as life processes. Examples are like cell can do respiration, cell can do digestion, a cell can do growth, a cell can control and coordinate, a cell can reproduce, and a cell can excrete, etc. These are the functions cell can perform. And so when a cell can perform this function, the cell become alive. Okay, a cell become alive. And when cell become alive, tissue becomes alive, then organ becomes alive, and likewise whole organ system and the whole organisms become live. That means If cells are working, then the organism is alive. What if the cell is not working? If a cell do not work, the organism is dead. It will die. Organism will die. That is why we call the cell structural and functional unit of life. Okay? Clear, everybody? Yes. Okay. Now, this can be the 2-mark kind of questions. Okay, so you can write the questions like why the cell is called structural and functional unit of life or organism. And then you can try to write the answer in your own proper words. But this is the answer. Okay. Yes. Now, we observed onion cells only under the microscope, but not only onions. Okay, every organisms we observe around us, they all are made up of cells. Okay, each and every living organism is made up of cell. And there are two type of organisms are there according to, okay, number of the cells organizing their body. Means how many cells are there in their body. According to that, we can see two types of the organism. Type number one is called multicellular organism. Okay, let me outline it for you. So number one is called multicellular. Now multi means many. Okay, what do you mean by multi? Okay, multi means many. But what many? Many cells are forming the body of organism. Okay, many cells will form the body of organism. It is called as multicellular. For example, okay, onion, fungi cell, fungi, plants and animals. These are the examples of multicellular organism. Now, multi-cell organisms are big in a size. We can see them with our naked eyes. Okay, remember that. And so we call them multicellular. Now, this is one more type which is called unicellular. Okay, this is unicellular. Uni. So, uni means single. Uni means single. That means their body is made up of single, one cell only. Means there is one cell and that one cell is the whole organism. For example, Paramecium, Euglena, Chlamydomonas, and bacteria. These are the examples of unicellular organisms. Okay. Now let's move forward. This is the activity we have to see. Let me. Okay. Now, the visuals are here. What is happening normally? For example, there is a sperm and egg. Now, if you don't know, I will tell you. The sperm means the sperm will coming from the male. And what a sperm is? What a sperm is? What is this? These are gametes. Okay. What are these? They are called gametes. Now, so sperm and the egg are gametes. Okay. Gamete cells. So sperm is coming from the male, or it is called male gamete. Okay. And the egg is called female gamete. Okay. So when male gamete and the female gamete, they fuses together. Okay. It is known as fertilization. Okay. The process is known as fertilization. If you are confused that what is happening, I will give you the story. Okay, that we are talking about sexual reproduction. Okay, we are talking about sexual reproduction. In sexual reproduction, sexual means there are two partners are present. There will be one male and there will be one female. A male will give its gamete called sperm and the female will give its gamete called egg. Okay, and you can see the sperm and eggs are here. in this big diagram. Okay, yellow color is sperm and the green color is egg. So when sperm and egg mixes together, fuses together, that process is known as fertilization. It is called as fertilization. And after the fertilization, okay, it forms another cell. That cell is known as zygote. Okay, that cell is known as zygote, the first cell of life. Zygote is called the first cell of life. And from that first cell, that one zygote cell undergoes multiple times cellular division, and from that one cell it will produces many cells, like you can see in this diagram. Okay, this is the one cell. For example, if I give you this arrow, okay, this is the arrow you can see. Okay, this is the zygote. Okay, the zygote is a single cell. That single cell undergoes, you can see, multiple times cellular division, and it will give me, for example, over here I can see 6 cells. Okay, so one cell undergoes cell division and give me many cells, and that is how we all are made. Okay, we all are made in the same process with the help of cell division. So this process is very important. Okay, this is very important process. Now let's see the topics. A zygote is a single cell. Okay, see, I'm reading from this here, written slides. Okay, so pay attention to the slide and the lines. Zygote is single cell. What a zygote is? A single cell, one cell. And that cell is coming from fertilization. And zygote is the first cell of the life. You can see when sperm and egg are fusing together, the first cell is zygote. And from the zygote, it will undergo the cell division. Okay, it is undergoing the cell division and giving us multiple cells. So it is the first cell of the life for multicellular organism. For multicell organism. Now, zygote can be seen in multicell organisms only, and that too in sexual reproducing organism. Okay, it cannot be seen in unicellular organisms or asexual reproduction. Okay, now through the cell division it will produces millions of cells, and that is how a multicell organism is made from a single cell. Okay, so from one cell it will make a big multicellular structure. I hope that this point is clear. Now, this is the kind of visual. You can see this is one cell. Okay, this is one cell. And that one cell undergoes cell division. Let me remove the drawings first. Okay, you can see this is one cell. This one cell first makes 2 cells. One cell undergoes the division and makes 2 daughter cell. And that 2 cells again undergoes the cell division and forms 4 cells. You can see 4 cells. And such a process every time, okay, repeated by the cells, and that is how this is the result. You can see this is a girl present in front of you. Okay, so that is the body of organism made by the cell through the cell division. Okay, so let's read this. We can try preparing temporary mounts. Now, mount means preparing a slide. So we can prepare a temporary slides of leaf peel. Okay, we did with the onion peel. The same thing we can do with leaf peel, roots of the onion, or even the peel of onions. Means there are many things we can do of a different sizes. Then after performing above activity, let us see what is the answer of the following questions. So question number 1. Do cells look alike in terms of the shape and the size? Means, okay, this is the thing. Means, do all the cells are looking same, exactly same in different structures, like cells of the root, cells of the onion peel, and the cells of the leaf? Do all the cells are looking same? Do they have a same size? Do they have a same shape? Answer is no. Okay, different cells of the different places are having different size, different shape. Then, do all cells look alike in the structure? Yes, their basic structure is almost same. Okay, the basic structure is same. Now, what is the basic structure? The basic structure is we draw previously. Okay, one outer line which is called cell wall. Inside the cell wall, cell membrane, then cytoplasm and nucleus. This is the basic structure. It will be same for every organism and every cell. Then could we find differences among the cells from different part of the plant body? Obviously, yes. Different cell from the different part of the plant will be different from each other. Okay, their functions also might be different. Now, what similarity could we find? What is the similarity? I just told you the similarity. Okay, their basic structures will be same. It is the similarity. Okay, I hope that you are able to answering these questions and understand the questions. Okay, if you can't understand, you can leave a comment. Okay. Now, what is this? These are different type of the cells from different organisms. Okay, like some organisms can also have cells of different kinds. Means a single organisms are made up of different type of cells. Like this is all, for example, human cells. Now, in human cells, you can see this is the RBC. You can see, yes, it is undergoing the animation. It is, okay. What is this? RBC, red blood cells. It's a blood cell. Now, blood cells are looking, circle, disc-shaped structure, which is your blood. Then this structure, what you see is, let me show you the structure. It is here. Okay. What is this? What is this? This is neuron cell. It's a neuron cell. Okay. Which is ultimately making your brain and your nervous system, with which you are able to control and coordinate your every actions. So it is the cell. And this last cell is any other simple cell which will make your body, okay, a tissue or something like that. Now, you can see all the three structures. Do they look same? Do they have a same size? So answer is no. All these cells are different sizes. They are of different sizes and they are looking different. Okay, they are literally very different than each other. Okay. Yes. So this is the thing you have to understand from this one slide. Now we will be moving ahead. Okay. Cell has some capability. Okay. Our cells have some capability. They can do something. Now what they can do? As the functional unit, the cell can perform. What it can perform? So see, the cell, you can see the first thing in the blue. As a functional unit, the cell can, it has capacity to perform certain basic function. The cell can perform certain basic function. Which function? Remember, respiration, growth, excretion, okay, digestion, etc. These are the function a cell can perform. Then, characteristics of all living forms. A cell is what? A cell will give, means the body characteristics. Okay, means different organisms, different body, they have different cells. Okay, then life possesses like growth, excretion, control and coordination. I'm so sorry. Yes, it can do the function which is called as life processes. Okay. Now let's move ahead. As a functional unit, the cell can do something like it has capacity to perform certain basic function. Now, which basic function cell can perform? You can see it can perform life processes like growth, excretion, control and coordination, etc. Digestion, respiration, etc. Okay, cell can perform these functions, and that is why cell is functional unit. And there are two type of cells we can see. There are two type of cells. Cell number one is called eukaryotic cell, and another type is prokaryotic cell. Let's have overview meaning of both the cells. Eukaryotic cell shows division of labor. Functions are divided by the cell organelles. In eukaryotic cells, what is happening? That a cell will do not do all the functions by itself. It will divide all these functions. Again, the question is which functions? The functions like growth, excretion, okay, respiration, like this, nutrition. So these are the functions a cell will do. When it is a eukaryotic cell, in a eukaryotic cell, these functions are divided, okay, among the cell organelles of that cell. Cell organelles. Okay, we have to learn about the cell organelles after this. So right now, remember the term cell organelles. Means in eukaryotic cells, the functions are divided. The one line is functions are divided among the cell organelles of that cell. And such cell is eukaryotic cell. That means division of labor is seen into eukaryotic cell. It is like you are alone in your home and you have to do all the functions. Like you will be cleaning your home, you will be making the food, you will be eating the food, okay, and then you will be washing your utensils, like that. So when such a situation is there, okay, we can compare this situation with prokaryotic cell. In the prokaryotic cell, a cell has to do all the functions by itself. Okay, there is no one to help that cell. That cell has to do all the functions by itself only, because they do not have membrane bound organelles. Because as I just told you that you are alone in your home and you have to do all the functions. Okay, so the same way the prokaryotic cell has to do all the functions by itself. The reason is alone. They do not have any membrane bound structures. But the eukaryotic cell is, you are living with your family. Okay, you are living with your family, means you are not alone. You will be doing some function, your sister, your brother will be doing some other functions, your mother and father will be doing some other functions, and together you all are doing the function, the chores of your home. So that is called division of labor. Okay, so division of labor is seen into eukaryotic cell, and the division of labor is not seen into prokaryotic cell. Let's move forward. Now, in multicellular organisms, similarly, okay, try to understand and compare. Same like eukaryotic cell, in multicell organisms also, the functions are divided. Like we have heart. The heart's function is to pump the blood. We have lungs like this in the image you can see. The lungs function is to respire, breathing. So lungs will be doing only breathing and not circulating. And heart will be only circulating the blood in the body. Then liver's function is to help in digestion. Then this is the kidney. The kidneys will help in excretion only. And the digestive system in this image you can see, it will be digesting the food. Now, all these organs are doing their own function, their own separate one single functions. And that is how the whole body is doing together different functions. This is called as division of labor. So understand, in multicell organisms, such type of division of labor is also seen. Okay. Now you have to do one activity. Okay. Try to differentiate between eukaryotic cell, prokaryotic cell, and multicellular organism and unicellular organism. Try to write this difference between these two, and you will understand the exact meaning. This is the same thing as I just told you now, but we have to read this. Okay, these are the lines from your textbook, dear students. So do not think that we don't want to see this. Okay, this is your textbook NCERT, line by line. I can understand that this video can be a little bit boring for you, but it is very important to go line by line. Okay, I'm trying my best to explain it in very simpler way. Now let's start. Similarly, division of labor is also seen within a single cell in many cases. Okay, I just explained it previously also, that if there is a single cell, in a single cell also division of labor is seen, and in multicellular organism division of labor is seen. Are you getting the point? Okay. In fact, each such cell has got certain specific components within it, known as cell organelle. Now the question is how a cell can divide its all the functions. So it can divide the function because inside the cell, it has few small little structure. These structures are called as cell organelles. What is this? Cell organelle. Now you can see what a cell organelle is. You can see in this diagram. Okay, this is the diagram. In this diagram, this is, let me give you overview here. Now, if you can see this circle in a center is nucleus. It is surrounded by this structure which is called as RER and SER, endoplasmic reticulum. Then this structure, you can see this structure, it is mitochondria. This is a vesicle, okay, or a lysosome. They are also vesicle. This is, do you see? Do you recognize the structure? It is Golgi body. So you can see Golgi body, ER, mitochondria, vesicle, this is centriole, this is nucleus. These are the structures present within the cell are known as cell organelles. They are what? They are cell organelles. These cell organelles are inside the cell. And these organelles are doing the function, different functions on behalf of the cell. So cell is doing the function? No. Cell is not doing the function. But instead, the cell organelles within the cell is doing the functions for the cell. And this is the reason we call it division of labor. Now, what are the functions? We have to see each and every functions in detail. Okay, so I will not be giving the overview over here. Yes. Now, let's read. Each kind of the cell organelle perform as a special function. Now, this is the point. Each organelle will be doing one function only, special function, the given function. Then making a new material in the cell. For example, it will be doing like making a new material for the cell, or it can be the cleaning up, means cleaning up the waste material from the cell, or it can be making energy for the cell. Let me give you example again, like the ER and Golgi body. Like, okay, first ER. The endoplasmic reticulum will be making some material. It will make protein, it will make lipid for the cell. It will be making only, nothing else. Number two, like this is the Golgi body. The Golgi body will be modifying the materials. It will modify protein, modify lipid. So it is doing modification. And second, transportation. Okay, one specific function. It will be doing nothing else. Then the mitochondria, powerhouse of cell. Now you know the reason why we call it powerhouse of cell. Okay, because it is producing ATP. It is producing ATP. So likewise, the functions are divided, and they will be doing single function and doing nothing else. A cell can live and perform all its function because of this organelle. Okay, why a cell is able to do all the functions? Because cell is not doing the function, it is the cell organelle doing the function. Now these organelles together constitute a basic unit called cell. So what a cell is basically, now for us the cell is, you can see in the diagram what a cell is. A cell is, I'm drawing it. Okay, see, the cell is having this outer periphery. Okay, outer periphery, which can be cell wall or plasma membrane. Then in the center it has nucleus surrounded by these small cell organelles and the liquid content of the cell cytoplasm. Okay, so do you see, a cell is consist of all these things only. So what a cell is basically, a cell which contains these organelles. That cell is doing some function, but that function is done by these organelles, and that is how when organelles are doing the function, a cell is doing the function. And when cell is doing the function, tissue can do the function, organ can do the function, organ system can do the function, and the organism is functional living. Okay, I think it is enough explanation. I will not make you bore more. Okay, now the chapter starts over here. Okay, from here you have to pay attention. A theory changes, question changes, basic structure of the cell. Do one thing. If you are making the notes, make a new point. Okay, write basic structure of the cell. This can be 2 to 3 mark separate question. Okay, describe the basic structure of the cell. Let me give you the question. Okay, what is the question? Describe basic structure of the cell. This is the question, for example. Clear? This is the question. So how to write this question? Now pay attention here. If we study a cell under the microscope, now you know how it looks under microscope. Okay, so if we study the cell under the microscope, we would come across 3 main features. And I showed you this also. Okay, so basically we can see 3 structures in the cell. Now what are the 3 structures? Structure number 1, plasma membrane. Now what a plasma membrane is? Plasma membrane means, let me take a pen for you. Yes, a plasma membrane means this outer structure of the cell. Am I clear, my dear students? Okay. Now, structure number 2, what we look at is the nucleus. Now, where is the nucleus? You can see the nucleus is here in a center of the cell somewhere. So that is the nucleus. And now there is empty space in between these two. Okay, that is hole getting filled by. It is getting filled by. Remember? Okay, just a second. It is now getting filled by like this. What is this? Cytoplasm. This is the cytoplasm. Okay, so now one point is very clear. We can see 3 basic structure into the cell. Outer periphery called plasma membrane. In the center of the cell, there is round oval mass called nucleus, and there is a liquid filled in that is called as cytoplasm. In each and every cell, we can see these basic structure, no matter what cell you are looking at. Okay, no matter what cell you are looking at, but these 3 basic structures are always present into them. Okay, now it's very important that anything here, anything less than this can not be called as the cell. Okay, you cannot call it a cell if there is not present these three things. For example, if there is plasma membrane is present, cytoplasm is present, but there is no nucleus. Now, there are some exceptions, okay? Let me give you, there are some exceptions, obviously, but normally we do not call it a cell. Okay? Cell means it must have all the three structures, but a science and biology always have some exceptions, okay? There are few exceptions, okay, like rbc of the human, rbc of the human, it do not have, it do not contains nucleus, okay. Similarly into xylem and phloem cells, okay, there are few cells, they do not have nucleus, okay. Then we have a virus actually, but to the virus we do not call a cell, it is not living thing. But remember that anything less than this, these three structures are not cell. So cell means having these three structures. And if they don't have this structure, then it is not the cell. Okay, I am hoping that I am very clear at this moment. Now, all activities inside the cell and interactions of the cell within its environment are possible due to these features. Let's see how. Okay. Cells is making contact with the outside environment as well as inner side environment. One organelle is connecting with the other organelles. One cell will connect with other cell. Okay, one tissue will connect with other tissue, one organ will connect with the other organ, so there is always connection, and this connection is possible actually, that this is possible, and that is why we are living organism, understand that. But the question is how, and we are going to learn that right now. Okay, so let's move. Okay, so this is the comparison between animal cell and plant cell. It's a comparison that whether these three structures are present or not. Like this is the animal cell, this is plant cell. Now you can see what they have is, just a second. Yeah, you can see in animal cell and in a plant cell, they have a nucleus. You can see the nucleus. Okay, a nucleus is always there. Okay, this is the nucleus of plant cell, a nucleus of animal cell. Means both cell have the nucleus common point. Then plant cell have the cell wall. Plant cell has the cell wall. Animal cell do not have the cell wall. Okay. But, and this is the plasma membrane. Animal cell have the plasma membrane. Plant cells also have plasma membrane. Okay. So plasma membrane, nucleus, and one more point, cytoplasm. So cytoplasm is present here also. And this portion liquid-like structure is also cytoplasm. Okay. So in every cell, we can see these three basic structures. Okay. I think this point is also very, very clear. Okay. Now, let's compare between plant cell and animal cell. Differentiate between them two. So, do plant cell have the nucleus? Let me take some here. Okay. So, plant cell have cell membrane. The first point is cell membrane. Okay, I can't. Oh. Just a second. I think you are not able to see a same color. So I will be changing the color over here. Great. Now, so do plant, see the first point is cell membrane. Now, do plants, this is the plant cell and this is the animal cell. Now let's compare. So cell membrane. So do plant cell have the cell membrane? Yes. Do animal cell have the cell membrane? Yes, both have the cell membrane. It's a common point. Then cytoplasm. Yes, both cell have cytoplasm. Then cell wall. Do plant cell have the cell wall? Yes. But do animal cell have it? Just okay. Do animal cell have the cell wall? Answer is no. It do not have it. Then nucleus. Yes, it has. And animal cell also has the nucleus. So that means both cell have nucleus. It is again the common point. Okay. Then endoplasmic reticulum. Yes, both cell have endoplasmic reticulum. Both have mitochondria. Both have Golgi body. Plastids. Only plant cells have the plastid. Animal cell do not have the plastid. Okay. Animal cell do not have plastids. Then, so here, let me write no for you so that you can not be the confusion. Okay. So no, animal cell do not have plastids. Then chloroplast. Again, animal cell do not have the chloroplast, but plant cell have the chloroplast. Okay. Then ribosome. Yes, both cells have ribosomes. Okay. Both the cells have ribosomes. Plant cell do not have the centriole and animal cell have the centrioles. Okay. Now you have to write this in a proper sentence as you are making the notes. Okay. And then you have to compare that how many points are there which are very, very common. Okay. I will be making a line for you. Then vacuoles, actually both have the vacuoles, but normally what we say is plant cell have the big vacuoles. Big vacuoles. And animal cells sometimes they have a vacuoles. If they have the vacuole, it is very, 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 very small. Okay. But in some, you can see it is very, very small, negligible. So let me write for you that is no. Sorry, not the no. No. Some cells do not have, animal cells do not have the vacuole. Just a second. Okay. So now see what are the differences between the animal cell and plant cell and what are the similar points. Okay. And this is it. Okay. Okay. Yes. 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 So let me remove this. Okay. So this is the comparison between the two cell and let's move ahead. So sorry. Yes. Now let's move ahead. Okay. This is the animation you can see. Okay. Of the now these are the few cells. Okay. That how now this is oh my mistake. Now this you see is the plant leaf cell. What is this? It's a leaf cells. Now you can see there are two type of cells. The shape is different of the cell. Okay. This is the palisade cells which are elongated and these are the mesophyll tissue which are circular, round to oval in a shape. And this is outer epidermal tissue or a cell which are flat structure. Okay, which is the flat structure. So you can see as the structure changes, the cell shape and the size also changes. Now, just a second. Let me. Okay, so Now let's move towards the next topic, that is plasma membrane. Here, okay, why plasma membrane? Yes, why plasma membrane? So, because let me draw it for you. In a cell, we can see three main basic structure. The very outer structure is plasma membrane. Then inside the center of it, nucleus. And this surrounding structure is cytoplasm. Okay. So now, as we are learning the cell, we have to see all these structures turn by turn. So right now, very first, we will be seeing the plasma membrane, its structure, function, okay? And then nucleus, then cytoplasm, and then organelles and cell wall also. Yes. Okay. So let me remove the drawing and let's start. Okay. What a plasma membrane is, okay? Pay attention now. Yes. This is the outermost covering of the cell. What is this? It is the outermost covering of the cell. You can see in this diagram. Do you see this diagram? Okay. The very outermost structure is what? Plasma membrane in animal cell. Okay. In animal cell, it is the outermost, and in plant cell, it is the inner second one. So this is the outermost covering of the cell that separates the contents of the cell from its external environment. Okay. So plasma membrane, let me put the sentence for you. Okay. Here. Outer into animal cell. Plasma membrane is outer structure of the animal cell. And in plant cell, see, I am writing over here, okay? In a plant cell, it is inner. Then what is outside in a plant? Cell wall, okay? Outside, cell wall is there. And inside the cell wall, plasma membrane is there, okay? That is the reason. Now, what it is doing? It is making a barrier from its external environment and its internal environment, so that the outer things, external environment, do not, okay, interfere the internal environment of the cell. It is its main function. Second, the plasma membrane allows or permits the entry or exit of some material, because it is very outside structure. Okay. A cell requires, see, we are eating, we are respiring, okay, we are excreting. Now these structures, these things are going inside the cell and coming outside from the cell. So they are going and coming through the cell, crossing the plasma membrane, okay, by crossing the plasma membrane. So plasma membrane is the structure who will allow this material to cross it, or it will stop this material to cross the cell. Second, it also prevents movement of some other material. It will allow some material to enter inside, and it will not allow some material to enter inside or exit the cell. Like what should enter inside the cell? Okay, before that, let me see you the 4 point, show the 4 point. The cell membrane, therefore, is called selectively permeable membrane. Now, understand this. A cell requires few things. Now, the question is, what cell requires? What cell requires? Yes, a cell requires food, water, and oxygen. Remember this? The cell requires food, water, oxygen, which is not there inside the cell. And that is why food, water, oxygen should be going inside the cell. Okay, cell will take this from outside. Now, what if plasma membrane will not allows this structure then? Okay, so this material must enter, and plasma membrane will allow these materials. Then what materials should come out from the cell? So it is the waste material will come from the cell, and CO2 will come out from the cell, which is called excretory material. It should come out from the cell. Okay. So these are the materials will cross the plasma membrane. Okay, yes. I hope that you are writing together. Okay. Yes. So it is the outer layer in animal cell. Okay. Plasma membrane is outer layer in animal cell and inner layer of the plant cell and fungi cell. Why? Because outside them cell wall is present. Okay. Yes. Clear? Shall we go ahead? Okay. Just a second. Yeah. Now this is the demonstration of material going and coming through out and in of the cell. Yes. So how? See the question. How does the movement of the substances take place into the cell? And how do substances move out of the cell? Okay. One thing before this we saw that few things plasma membrane will allow few materials to enter inside the cell or exit the cell. Means something will come in a cell, something will go out from the cell. The question is how they will come in, how they will go out, the method. Okay. So that is known as diffusion. Okay. That is known as diffusion. Now this is the explanation of the diffusion. Let's read line by line so that you can understand. Okay. Cell requires oxygen and water. Okay. What cell requires? Oxygen and water from outside to inside the cell. Then it needs to remove the waste material like CO2 and nitrogenous waste from the cell into outside. Okay. From the cell to outside. So this is the main requirement of any cell. Okay. If this do not happen, cell will die. Without food, without oxygen, cell will die. Or without excretion, cell will obviously die. Now, some substances like carbon dioxide or oxygen can move across the cell membrane by a process called diffusion. Okay, so how these things will move across the membrane, that is called as diffusion. The process is called as diffusion. So these molecules will move through the process called diffusion. Now let's define the diffusion. It's a spontaneous movement of a substance from a region of its higher concentration to its lower concentration. Means they will be moving from where they are high to where they are in low. Like see in the diagram. Okay, I will show you from this diagram here. Yes. Now you can see over here this blue dots. Just count the dots. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 dots on your left side. Okay. 12 blue dots. And on your right side, there are 6 dots. That means on your left side, there is high concentration of something. And on your right side, there is low concentration of something. So these materials will move from left to right side. Left to right side. Why? Because it is high this side and it is low this side. Clear? So you can explain this in another terms like it is the movement of the molecule. It is the movement of the molecule from its higher concentration to its lower concentration. Means they will be moving from where they are high to where they are in low concentration, in low amount. Clear? And this movement will continue till both side becomes equal concentration. It will stop, but when becomes equal at the both side, when left and right both sides are almost equal, at that time this movement will stop. And this process, the movement is known as diffusion. Clear, students? Am I clear? Yes. I hope that you are getting these points. Okay. Because if you don't understand this, the next sides you won't understand in better way. But I have few examples, okay, for you. Don't worry. Now, this is called as concentration gradient. Now, this thing, what this? At one side, something is high, and at one side, something is low. This is the difference is called concentration gradient. Understand that? Concentration gradient means at one side is high, another side it is low. This difference is called concentration gradient. So this is how the material can cross the membrane according to its concentration gradient. Clear? They can only move according to concentration gradient, like from higher to lower only in diffusion. Yes. Okay. Now, see, for example, this is the visual for you. Like oxygen is low inside the cell. Okay, try to see this. Okay, very carefully. Yes. Now, there is, see, oxygen is high outside the cell and oxygen is low inside the cell. Okay, why? Because obviously outside air is present and oxygen is very high, and its requirement is inside the cell. So inside cell will utilize the oxygen, and so it is low. So outside high oxygen, inside low oxygen. So oxygen, because of this concentration gradient, oxygen will come from outside to inside of the cell. Clear? How oxygen is able to cross the membrane? Okay, through the diffusion, because it is high outside and low inside, and according to the definition, anything will move from its high to its low. So oxygen will enter inside the cell, then reverse. After the respiration, they will cell is using the oxygen, it will make carbon dioxide. Now that carbon dioxide is very high inside the cell. It is high inside the cell, okay, because cell is producing it continuously. Okay, and outside the cell it is in low concentration. So carbon dioxide will exit the cell through the diffusion. So it will diffuse out. Oxygen diffuse in because it is going inside. So diffuse in, and carbon dioxide diffuse out. Exactly. Now, see very carefully. You will be seeing the animation over here. Okay. Clear. Now see. Okay. See this. Now, as oxygen will enter inside the cell, oxygen becomes high because oxygen has diffused inside the cell. So inside the cell now oxygen is high and carbon dioxide is coming outside. Clear? Okay. Yes. Okay. Continuously this is happening. Just a second. Yeah. So now similarly, water is high outside and food is high outside the cell, and they are required and low inside the cell. So food and water both enter inside the cell, and that is how water becomes high inside, food becomes high inside. And once they become high, it will stop the process of diffusion. Similarly, now the one more example of the diffusion is how we are able to no respire, breathe. So you can see the visuals over here. Like oxygen is high outside and oxygen is low in your lungs, whereas carbon dioxide is high in your lungs and low outside. And that is why oxygen will come from outside to inside and carbon dioxide will exit your lungs. Okay. through the diffusion. Yes. So is it clear? It is clear. Yes, very good. Now this is one more example in a different with a different example of a diffusion. A same thing. Okay. Like you can see in this diagram. First, let's see the diagram. Okay. At one side, let me, okay, there is a given. This is region A and this is region B. There are two different sides of one place. Region A is what? Region A is showing higher concentration. Region A shows higher concentration. And this is region B, which shows low concentration. Why? Why low? Why high? Count the Na plus. Over here, 9 Na plus is there in region A. Whereas in region B, you can see only 5 Na plus is there. So basically it is low and it is high. And that is why Na is high over here and Na is low that side. Okay. Okay. Now one more point. Whenever there is concentration is higher, water is always low. So when there is higher concentrated area, the water is low concentration. And when there is low concentrated area, water is always high. It's a reverse thing. Why? I will explain it. Okay. So, but remember, water is, let me write. Okay. Water is low. And here, water is high. Clear? Na plus is high, but water is low. And region B, Na plus is low, but water is high. It is always the case. Yes. Whenever water is having something in big amount, okay, dissolved by something in very high concentration, that means water is always low. Now, why? I will try to explain it. Okay. I have a solution for that. So don't worry. But right now, see this example. Okay. So region A, high concentration of Na plus. Region B, high concentration of Na plus. Sorry, low concentrations of Na plus. Yes. Now, according to the diffusion, what should happen? According to the diffusion, what should happen? Write your answer into the comments. Okay. Write your answer into comments. What should happen? Na plus should go from higher to lower. Means Na plus should move. Na plus should move from region A to region B. This should happen according to the diffusion. But you can see the difference that this both area are separated by this plasma membrane. You can see this is the plasma membrane. This is the plasma membrane. So plasma membrane is dividing both the structures. And because of that, now Na plus is unable to cross the membrane. It can't cross the membrane. So when Na plus cannot cross the membrane, in such no situation, now water will move from its higher concentration to its lower concentration. Now, remember where water is high and where water is low. Remember, water is high in region B and water is low in region A. That is why water will move from region B to region A. Now, why this happened? Because Na plus simply cannot cross the membrane. If they could cross the membrane, Na plus would have crossed it. And so Na plus can show the diffusion. But under the circumstances where Na plus cannot cross the membrane because of the plasma membrane is present. And plasma membrane is a selective permeable membrane. It will not allow the Na plus to cross. And that is where it's a problem. What is the problem? The problem is at one side there is high concentration of Na and at another side low concentration of Na. And such imbalance is not good for the life. That is where life will try or cell will try to make it under the balance. where it will try to lower the concentration. Now, how they will lower the concentration? Very simple. If Na plus cannot cross, water will cross. Okay, so water is crossing from its high to its low, and that is how more water into more Na plus, that is how becomes equal concentration. Enough of this. Let me click over here. Now, this phenomena is known as osmosis. What is this? What is this? Okay, what is this? Osmosis. It is not a diffusion, it is called as osmosis. Means when water will move from its higher concentration to its lower concentration, it is known as osmosis. Okay, very good. Yes. So, see, osmosis is the passage of the water from its region of high water concentration through a semi-permeable membrane to the regions of its lower water concentration. Means, basically, I can say that this is the diffusion of water. In one line, what I can say is, it is the movement of water from its higher concentration towards its lower concentration. Over here, water is moving, means diffusion of water. It can also be seen as water movement from lower concentrated area to higher concentrated area. Yes, just a second. Okay, let this sentence finish. Okay, now again. Okay, I hope that I have not made you confused over here. Okay, I will try to uh, make it very simpler for you. Again, now what we are learning is difference between the diffusion and osmosis. Remember first, what is diffusion? What is diffusion? Diffusion means Na plus when move from its higher concentration to its lower concentration, means when any molecule move from higher to lower concentration, it's diffusion. But when molecules can't cross the membrane because the membrane is present in between, and because of that, water will move. Now, water will also move according to the same concentration gradient from higher water to lower water. Okay, so see again, we are reading. Osmosis is the passage of the water from a region of high water concentration through a semi-permeable membrane to regions of lower water concentration. I can also write like it is a diffusion of water. I can call it diffusion of water because same water is going from higher to lower. Now, it is a movement of water from its higher concentration to its lower concentration. I can say in another words. Okay, don't get confused with these similar kinds of words. They are actually looking similar, but they are not similar. Okay, yes. So from higher to lower of water, or it can be also seen as water movement from. Now, the sentence is changing over here. This is the changed sentence, okay? Pay attention. Water movement from lower concentrated area to higher concentrated area. This is the reverse, okay? From lower concentrated area, not concentration. Concentrated area to higher concentrated area. Now, see the diagram. Which region is called higher concentrated area and which region is called lower concentrated area and why? See, region A. Region A is having so many Na plus. Sodiums are there. So that is why this region is called higher concentrated area. And region B is having only 5 Na plus than region A. It's called lower concentrated area of Na plus. Okay. So this is no area wise concentration we are just saying. So over here, The water is, but the water is moving reverse. The water is moving from region B to region A. Remember? Region A to region B. No, region B to region A. Means from lower concentrated area to higher concentrated area. Let me see. We are taking time so that you can just a second. Yes. Okay. So you can understand the core idea. Remember that. Okay. This is lower concentrated area from to higher concentrated area. Okay. Water is going reverse from, let me give you arrow. Okay. See, from lower to higher concentrated area. And what is going? Water is going, not molecules. Molecules of water is going, which is also known as diffusion of water. And you see why, how and why it is happening. Why water is going from its lower concentrated area, sorry, from lower concentrated area to the higher concentrated area? Because answer lies downside. You can see the answer. And what is the answer? Answer is here. Because water is high in the lower concentrated area and water is low in higher concentrated area. And that is what we are saying, that water or any molecule always will move from its high to its low. Clear? Okay, yes. Very good. Very good. Very good. Okay, so let's move now to the next slide. What will happen if we put an animal cell or a plant cell into a solution of a sugar or a salt? A next activity. This is the activity. Activity of osmosis and the diffusion. The question is, what can happen if I put the animal cell or a plant cell into water? Different type of water solution. Okay. In a life, many animals are also living in a water and they have plasma membrane. So think of the osmosis in respect of the situation. Like, see, we just saw, let me go big back side. Okay, what is happening? See, in here, osmosis. Osmosis means when you are surrounded by the water, water enter inside the cell. Or water will enter from its higher concentration to its lower concentration. Or the second definition, the water will move from higher, never higher. See, lower concentrated area to higher concentrated area. Okay, lower to higher concentrated area. Now imagine that there is a fish. A fish body is higher concentrated area and the water is lower concentrated area. Is it? Yes. So water will move from outside to inside the fish. Okay. Now fish is getting the water, continuously getting the water. And that is how the fish becomes swallow. Okay. It will swells up and then blast. But is it really happening in the situation? In reality, the answer is no, it is not happening. And the thing is, okay. So basically we have to learn osmosis. Okay. And before that, okay, let me make it bigger. Now we have to learn osmosis. Okay. And for the osmosis, there is a small activity or a practice you have to learn first. Let's learn osmosis. Okay. What is osmosis? Diffusion of water. Water will move from its higher concentration to its lower concentration. Now for this, there are one small experiment of the three glasses. Okay. I will try to explain it to you and actually you can do this at home. Okay. It's an activity. Let me write Okay, it is what? It is activity. Now, what is the activity? Activity is, now see very carefully. Take three glasses, okay? Or you can see, you can take the Petri plates, okay? So I am writing, sorry, drawing a Petri plates. This is what? Petri plates. You can take glasses also, doesn't matter. What is this? This is Petri plates. Okay, you can choose glasses. So, okay, let me take the glasses for you. Okay, so osmosis, we have to take three glasses. Let me, or a beaker, okay? Clear? Now, give these three glasses name. This is A, this is B, this is C. Clear? Now, in A, what we have to do is, all the three, we will be putting same amount of water. Okay, we have to put same amount of water into all the three glasses, like till here, okay, same amount of water. See very carefully and understand. So what do you see? We are pouring water in all the three glasses in equal amount. So equal amount of water. Clear? Equal amount of water. Now means there is no difference in all the three glasses, okay, except the name. There is A, B, and C, the name, and they have same amount of water. Now in this water, we have to put a cell. And instead of the cell, we can put grape, okay, resin, the dry grape. So, but before we do that, we have to understand the concept. Okay, so we have added the water. Yes, it is over. Yes. Now, second thing is, we need to put, we have to create concentration. Okay, for the concentration, we have to add, let's add salt. Okay, let's add salt. Salt. Now, this salt will be different amount. Okay, like in A, we will be adding, let me write this now. Okay, in A, I will be adding 10 tablespoon of salt. In B, we'll be adding 1 tablespoon of salt. And in the C, we'll be adding 0 tablespoon of salt. Means no salt at all. Now, this is the difference. Clear? After adding this, we will be adding one grape or a cell inside. So let me write it. Okay, this is the grape. For example, this circle is grape. It's a grape. It's a grape. It's a grape. I think the scenario is clear for you. Okay. How to understand the osmosis? Very easy. Take three glasses of same sizes, then pour same equal amount of the water. You can see it is added. Now give them name A, B, and C. Now add them with the salt. In the A, we are adding 10 tablespoons of salt, which is very higher. Then in B glass, we are adding 1 tablespoon of salt. And in the C, we are adding no sugar at all. And then we are adding one cell, or in place of the cell, we are adding grape. Okay, resin. Now, this is the difference. Now, one thing, if you know, and even if you don't know, you should know this now. So I will be I'll be saying you that there is a cell, and cell has a cytoplasm. Okay, what cell is having? Cytoplasm. Cell is having cytoplasm, and cytoplasm is always filled by some kind of salt, sugar, and few concentration increasing material. So cytoplasm of the cell is concentrated area. It is concentrated area. And that concentration area, we cannot change it. Okay, it will be remaining the same as it is, whatever it is. And that is how it is a concentrated area. But the thing is, we can't change it. And so what we are doing, we are changing the outside water, because we have to do the experiment of osmosis, where there will be water high at one side and water low at one side. That means one side higher concentration, one side lower concentration. And so we are adding 10 tablespoons of salt. Now let's jump onto the A. What is happening in A? In the A, because there is 10 tablespoons of the salt. Okay, there is 10 tablespoons of the salt. That means, okay, the salt is outside water. Okay, in outside water, salt is there. So that is why the outside solution is high. Okay, so sorry, higher concentrated area than cell. It is higher concentrated area than the cell, because there you can see there are 10 tablespoons of salt is there. But in another, in a class B, let's assume that there is equal to 1 tablespoon of sugar concentration is present into the cell cytoplasm. Okay, cell cytoplasm is having same equal concentration. So that is how the outside solution is equal concentrated. It is equal concentrated in B. Whereas in the C, because we have added nothing in the outside solution, so that outside solution is lower concentrated, because there is nothing in the outside solution, but cell cytoplasm has something. So outside solution, when we compare, it is lower concentrated area. Okay, is this experiment clear? Okay, so again I'm repeating the whole process. We'll take 3 glasses. Okay, then add equal amount of the water in. Okay, give them name A, B, and C. Now in A, we are adding 10 tablespoons of salt, means that it is very high salt in that water. Then in B glass, we are adding only 1 tablespoon of salt, which is nearly small. And in the C glass, we are adding nothing. Okay, and then we are adding one grape in each glass. Now grape is having already sugar inside, which is a concentrated area. Okay, which is equal in all the 3 grapes, it is same. So now I have to compare the cell, means the grape, and its outside solution. Okay, the cell and its outside solution in glass A, because there is so much salt, so outside solution is higher concentrated as compared to the cell. So that means inside the cell, it is low concentrated. Clear? And outside solution is high concentrated area. Area is high concentrated. Number 2, in glass B, there is 1 tablespoon of salt in outside solution. Okay, when we compare with the cell, so it becomes equal, kind of equal concentration. Okay, and in glass C, the outside solution is without any salt, means outside there is no salt present, but inside the grape there is some salt obviously present. So that is how the inside of a grape is higher concentrated. It's higher concentrated as compared to outside. That means outside solution is lower concentrated. Is it clear? In such situation, the A is known as, the outside solution of A is known as hypertonic solution. Okay, in glass A, the outside water is known as hypertonic solution. In glass B, because it is equal concentration, okay, it is called isotonic solution. And in C, the outside water is lower concentration than the cell. So it is called hypotonic solution. Okay, so why it is called hypotonic? Because it is having lower concentration than the cell. Why it is called isotonic? Because it is having kind of equal concentration than the cell cytoplasm. And why it is called hyper? Hyper means so much. Why? Because the outside solution of a cell, of a grape, is having much higher concentrated than the cell cytoplasm when we compare. Okay, that is how we are getting hypertonic solution, isotonic solution, hypotonic solution. Okay, and and now we have to apply the rule of osmosis. What is happening in osmosis? Remember what happens in osmosis? I will be writing here. Okay, what happens in osmosis? Water will move. Okay, water will move from high to low water. Okay, water always will move from its high to its low. And there is one more rule I told you, that whenever a concentration is high, let me write it. Okay, concentration is high, over there water is low, and vice versa. When concentration is high, water is always low in that region, and when concentration is low, that means over there water is high. Now apply this rule over here. Apply this, okay? Wherever concentration is high, water is low. So let me, okay. Now in A, what you can see, outside solution is higher concentrated, means concentration is high. When concentration is high means, concentration means high means, see over here, concentration high means water is low, means outside water is low and inside the grape water is high. Okay, so let me again write it over here. Okay, I'm writing over here. Inside of the cell, water is high, whereas outside water is low. Okay, and that is how over here, because it is equal, so let it make equal now. But in here, outside water is lower concentrated. When concentration is low means water is high, so that means outside water is high, whereas inside of the cell water is low. Now, is this point clear, that why the water is low and why concentration is high? Is this clear, one thing? Okay, so now just basically apply the rule. Water will move from higher concentration to lower concentration, or the water will move from high water to low water. Just simply compare where the water is high, where the water is low. So you can say and see that, see downside I have written. Okay, inside water is high means water is inside high and water outside low. So water from its high have to come where it is low. Okay, so water is coming outside, out of the cell. Okay, water is coming out of the cell. Then in hypotonic solution, outside water is high and inside water is low. So water will move inside. Water will move inside. Okay. Now, when water is moving inside, it is known as endo osmosis. It is known as endosmosis, okay, where water from its outside enter inside the cell. And when water is coming outside, it is known as exosmosis, means water is coming outside. And when exosmosis occurs, means when water is leaving the cell, cell will shrink. Cell will shrink. And over here, because water is entering inside the cell, it is like you are, okay, blowing a balloon with the air. When air enters into the balloon, what happens? Balloon blows. Same over here, cell will swells up. Clear? So this is the effect of all the three solutions: hypotonic solution, hypertonic solution, and isotonic solution. Now, isotonic solution, in isotonic solution, because concentration of water is equal, so a same amount of water enters inside and same amount of water exit the cell, and that is how there is no change in the shape of a cell, size of a cell. Okay, there is no change at all. Now, this is the thing I have explained, but we have to write it properly. Okay, so after this, there are 3 slides of all each hypotonic, hypertonic, and isotonic solutions. So let's jump and move to the next slide. I hope that you are getting this now. Okay, and if you can't understand, you can comment in the box so that later on I will do this experiment for you. Okay, I will make a separate video with the same experiment and I will put that for you so that you can see, visualize it with your eyes now. Now, before that, what you have to do is, just a second. Okay. You have to do one thing. Either you write this, okay, you have to write this in your notebook, or you can take the snapshot and later on you can write it. Okay, because I am going to erase this from this slide. Okay, now clear all drawings. Great. So the first hypotonic solution, let me remind you again, hypotonic means outside solution is lower concentrated. Yes, very good. Now see. Okay, if you have paid attention, okay, you should know this. If the medium surrounding the cell, means outside solution, has higher water concentration, what is written? Higher water, means when water is high, that means concentration is low. Okay, so higher water concentration than the cell, compared to the cell, meaning that outside solution is very dilute, means outside solution is very dilute than the cell cytoplasm. Yes, the cell will gain water. What is happening? Because outside water is high, it will enter into the cell. Okay, gain the water by osmosis, and such solution is known as hypotonic solution, means outside solution is known as hypotonic. Hypo means lower concentration than the cell. And when water enter into the cell, it is known as endosmosis. Okay, endosmosis. Now, water molecules are free to pass across the cell membrane in both the directions. Either it can go inside or it can come inside. Means it will come in the cell or it can go from the cell outside. But the thing is, how it will be moving? They will be moving according to concentration gradient. Means to move the water, there should be at one side higher concentration of water, at another side there should be lower concentration of the water. Okay. But more water will come into the cell than leave. Okay, but what is happening over here? Okay, more water is coming inside and small water is going outside. Okay, then, okay, yes, great. The net means overall result of the water is more entering than exiting. And that is known as endosmosis. And because of this situation, the cell will become bigger, swells up. Yes. Now this is the diagram. Petri plate is there. I have put one cell inside and there is simple water outside in the Petri plate. Okay. That means outside there is nothing. But inside the cell, there is some concentration present. So cell cytoplasm is concentrated. Okay. Cell cytoplasm is concentrated. Means inside the cell, low water is there. And outside the cell, high water is there. Outside the cell, there is high water. Yes. Yes. Please stop commenting right now. Yes. Okay. So now you can see because water is high outside, low inside. So water will enter inside the cell. Okay. Cell cytoplasm is concentrated and outside simple water. So water will enter inside the cell. Now, why outside water is simple? Because we have added nothing. You can read this. Yes, to the cell cytoplasm, this gets low concentrated region. Outside is low concentrated area and inside the cell, high concentrated area. Now, second situation, isotonic solution. Now, isotonic solution means, yes, now we have to read this. Okay, first, if the medium has exactly the same water concentration as the cell, isotonic solution. In this situation, when concentration is equal, okay, there will be no net movement of the water across the cell membrane. And such solution is known as isotonic solution. Such water is isotonic solution. The outside medium is isotonic. Iso means equal concentration. Now, in this situation, the same amount of water enter in the cell and the same amount of water exit the cell. So there is no net change into the shape and size of the cell. And the last one, hypertonic solution means very high concentration than the cell cytoplasm. When the medium has, just a second, let it all come. Okay, now let's read this. When the medium has lower concentrations of water than the cell, means outside medium is having high concentration. Okay, high concentration of something, molecule, and then the cell. Or in other terms, outside is low water and inside the cell, high water. Okay, do not be confused with these sentences. Okay, why? I just told you previously. Concentration is high. That means when the concentration is high, water is always low. And when the, okay, vice versa. When the concentration is low, that means high water. Okay, now apply these two lines over here. See, the medium has lower concentration. Medium means outside water, has lower concentration. See, when concentration is low, see here, when the concentration is low, means water is high. That means outside water is high. Outside high, see very carefully again. Outside is concentration high, the first one. Okay, so concentration is high outside. Okay, let me now, sorry, here, let me write here for you. It is the case of outside, so you do not get confused. And this is the case of inside cell. Okay. Now, so outside concentration is high. That means water is low outside. Outside water is low. So let me write here low water. Okay. And inside the cell, water is high. Why water is high? Because concentration is low. Okay. So basically what should happen? Water should move from its high concentration that is inside the cell to outside solution. Okay. So that is why the outside solution is known as hypertonic solution. Yes. And that is how the cell will lose the water by osmosis. This is known as exosmosis because water is coming out of the cell. So it's known as exosmosis. Because of that, cell will shrink. Okay. I hope that I have not stretched the topic. Okay. I'm stretching because of the few students that do not understand it. So, and if you can't understand, you can, okay, leave this and go to the previous, the single slide where I have explained all the things in very short. Okay, you can utilize that one only. Yes. Okay. Now, this is the experiment of the same thing. What is the same thing? Osmosis. Okay, this is the experiment of osmosis, but it is called as plasmolysis. Now, the question is, when we put the animal cell into the hypertonic or hypotonic solution, it will either shrink or swells up. It's a condition, the whole cell. But the question is, what happens if we put the plant cell into such solution? Okay, there will be no same effect. There will be no same effect because plant cell have the cell wall. Plant cells have cell wall. Okay, because of that. Now, first see the video. Okay, see, this is the video of peel of a plant leaf. It's a peel of plant leaf, and this last one is the peel of onion leaf. Yes. Now, this is the video where you can see the cell is undergoing the plasmolysis, means the shrinkage of cytoplasm. See, very carefully first see that video. Okay, I will explain it, that what it is. What it is, I will explain. In the end, do you see? If you don't see, I will again. Okay, now I will pause the video. Very carefully see. You see this area, okay? I will make an outline. You see this area, for example. This area, this area, this area, okay? Actually, any area. Very carefully see this area. Now the question is why? Because you can see it is full of cytoplasm. It is pink in the color. It's pink in color, cytoplasm. Okay, see. Now slowly, that cytoplasm, which is in very big area, will come to in a corner of a cell, in a very small area of the cell. See very carefully. Slowly, that area will turn into different looking structure. See that, huh? Very carefully see this. Do you see the changes? Yes. Do you see the changes now? Yes. Actually, the video is going a little bit up or down, but you can see the changes than the previous one. Okay. Yes. Now you see, okay, these circles which are right now upside, they are actually here at this region. Do you see? Do you see this cell? Do you see this cell? Okay. The whole cell is not covered by cytoplasm, but cytoplasm is shrunken into the corner somewhere in a cell. Do you see? Now, the same thing is very clearly seen here. first, this is what you look at is a cell. Okay, this is a cell. It's a cell. Okay, each hexagonal kind of looking structure is a cell. Okay, you see this is also the cell. And in that cell, previously cytoplasm was equally distributed. But right now, after plasmolysis, you can see it is just present in a corner of a cell. In a corner of a cell. Same thing you can see here. This is the cell. See the outline. Huh? Very carefully see this outline. Okay, cell. And this is the cytoplasm in center somewhere. Okay, shrunken. See, this is also shrunken cytoplasm. See, this is also like, okay, totally shrunken. So this phenomena is called as plasmolysis. Now the question is what it is and how it is happening. So we have to learn that now. Okay, I am showing you the video first so that you get some interest that what it is. Now, if you put the plant cell into hypertonic solution, remember hypertonic solution outside. Let me see, this is how it looks first. Now, let me write it on the same thing. I will draw it again for you. Okay, let me write actually. When you put the cell, okay, plant cell, where we have to put the plant cell into hypertonic solution. Now, this is the situation. We are putting the plant cell into hypertonic solution. Now, clear the idea of hypertonic solution. Hypertonic means outside solution is higher concentration. Higher concentration. That means water is low. So let me just write it. Outside solution is higher concentrated. That means water, so sorry, yes, water is low. Means outside water is low. And cell cytoplasm has high water. Okay, cell cytoplasm is having high water. So basically what is going to happen? Inside the cell, water is high, and outside the cell, low. So water will move from inside to outside. Okay, let me write it. So basically, water moves in to out. Means from the cell. Okay, moves from cell to outside solution. In one word, if I say this phenomena, then it is known as exosmosis. Exosmosis will occur. So when we put the plant cell into the hypertonic solution, water will come out of the cell. When water comes out, because water is coming out, the cytoplasm's water comes out, and cytoplasm losing the water, and so it become shrunk. Okay, and the more water comes out, okay, it becomes more shrunken condition, and so the cytoplasm will gather in a corner of a cell. But plant cell has the cell wall. You see this, I will give you outline. You see, this is this outline is what? It's a cell wall. This outline is what? Cell wall. Because cell wall is there, and cell wall is a very protective structure. It will not let the whole cell shrink. Only cytoplasm is shrinking. Only cytoplasm is shrinking because of the cell wall, the protective structure. It will hold the whole cell structure. Okay, and so only cytoplasm will undergo the shrinkage, and this phenomena is known as plasmolysis. This phenomena is known as plasmolysis. Okay, I am clearing the drawing first. Now let's move ahead. Okay, now we have Up till now, we have learned the plasma membrane. Okay. Plasma membrane. If you remember, we have to learn all the structures of the cell. So plasma membrane, one structure is over. Now, second structure is the center of a cell is called nucleus. Okay, it's a nucleus and cytoplasm. Two things are remain. So next is nucleus. What is the nucleus? Let's It was first discovered by Robert Brown. Okay. It was first discovered by scientist Robert Brown in 1831. Then it consists of two parallel membranes. I will give you the diagram also. Okay. It's a double membranous structure. And it is always present normally, okay, into the center of a cell cytoplasm. It is present into cell's cytoplasm, normally center. You can see in this back diagram. Let me move this picture from here. Okay, what do you see? It's a nucleus, and nucleus is somewhat center of a cell. Okay, this is cytoplasm. Yes. There is a gap between those two membranes, which is called as perinuclear space. Okay, there is a gap. Now, this gap you will see into the diagram. Okay, I will explain it. Then, within the membrane, it contains nucleoplasm. Okay, means cytoplasm of the nucleus. Inside the nucleus, okay, but now I will draw it for you. For example, this is the nucleus. It's a nucleus. And inside the nucleus, what we can see, cytoplasm. So this is that cytoplasm. Okay, I'm drawing the cytoplasm. This cytoplasm of the cell nucleus is called as nucleoplasm. It is a nucleoplasm. Clear? Yes. Now, nucleoplasm contains, now inside that nucleoplasm, it contains chromatin material, DNA, and a small structure, okay, that is nucleolus is present. And the nuclear membrane is interrupted at the various places, which is known as nuclear pore. Okay, it happens because of the fusion of the two membrane. I will show you. And this will create the pathway from where something can, from the cytoplasm, enter into the nucleus, and from the nucleus, something can enter into cytoplasm. Okay, it's a basic pathway. Now, what is entering and what is exiting? Some protein, RNA, okay, they will exit and enter from the nucleoplasm to the cytoplasm and vice versa. Okay, now you can write these points because after this, you are going to understand that from the diagram, okay? Okay, what is this? This is the very outer membrane of the cell. Which are the two membrane? Which two membrane? Let me give you the idea. One is this membrane, one is this membrane, and one is this membrane. Okay, so there are two membrane, outer membrane, inner membrane, and at certain point, okay, they are fused. So this is that fusion. It is that fusion. It is that fusion where both the membranes are getting fused, and because of that, you can see there is one passage. This is the passage, which is known as nuclear pore. Sorry, it's a nuclear pore. Okay, now next is within these two membranes, the whole portion is there. This is known as nucleoplasm. Okay, and in that nucleoplasm, it possesses this red color is nucleus DNA. It has the DNA, and this small dot is nucleolus. Chromatin. Okay, this red one is the DNA, chromatin material, and this pink one, small dot, is nucleolus. Great. Now, let's nomenclature it. This is a nuclear membranes. There are two membranes. This is nuclear pore. This is nucleoplasm. This is nucleolus. This is DNA or genetic material. Clear? So if you learn the diagram, you can write the whole theory. Yes. Now, on the basis of the nucleus, okay, or nuclear membrane, there are two types of organisms we are going to learn. Let's read. Types of organisms on the basis of presence or absence of the nucleus. Means if the nucleus is present or if the nucleus is absent. There are two types of organisms on the basis of the nucleus, presence or absent. Like first organism is called as eukaryotic organism. Okay, it is known as eukaryotic organism. So sorry. Eukaryotic organism. Now, eukaryotic means what happens? The genetic material is enclosed, bounded by the nuclear membrane. Okay, there is a nuclear membrane present. If you are confused, you might have draw that diagram. If you haven't drawn, then please, okay, go back in the video right now and see that diagram first. Okay, that what is called nuclear membrane. So the DNA is basically, let me draw for you right now. Okay, now this outside membrane is called nuclear membrane. This is the nuclear membrane, and inside this DNA or the genetic material is present. When such thing is there, this is known as nucleus. It is known as true nucleus. And when the nucleus is present into the cell, that cell is known as eukaryotic cell. Clear? So what is eukaryotic cell? The DNA is enclosed by plasma membrane or nuclear membrane. And such structure is known as nucleus. And when nucleus is present into the cell, it is known as eukaryotic cell with the true nucleus. And there is one more type of the organism called prokaryotic organism. Now, prokaryotic means what? Over there, their DNA is not enclosed. The genetic material is not enclosed by plasma membrane, nuclear membrane, basically nuclear membrane. So let me draw. Okay, for example, see the diagram. This is the DNA. This is DNA. It's a DNA. But DNA is not enclosed by the membrane. See, I'm not drawing this membrane. See, this is the membrane, which is called nuclear membrane. But I'm not drawing that, okay? It is not there. Clear? And this is the cell. It's a cell, plasma membrane. So DNA is not enclosed by the plasma membrane, nuclear membrane, and so it is not known as the true nucleus. Instead, it is known as nucleoid. It's known as nucleoid. That means the DNA or the genetic material is floating into cytoplasm of the cell, not okay, separated. And such cell is known as prokaryotic cell. For example, bacteria. For example, bacteria. You can see from the diagram, okay, the difference. Yes, exactly. So I hope that two things are clear, that what is eukaryotic cell and what is prokaryotic cell. Now, this thing, it is looking, maybe it is not fits in your space, screen. Okay, so let me make it small for you. Okay, over here we are going to learn. I hope that you are able to see this. Okay, or let me make it small. Okay, this is good. Okay, so this is the difference between prokaryotic cell and eukaryotic cell. Okay, it is a difference between the prokaryote and the eukaryotic cell. So in a prokaryote nucleus, DNA does not enclose within the nuclear membrane. It is naked. Okay, means nucleus is absent, you can write. Then in eukaryotic organism, DNA is enclosed within the nuclear membrane. It is known as true nucleus. So nucleus is present. Then nuclear membrane, nucleus, oh, so sorry, it's a name, nucleus name, what it is known. So in a prokaryotic cell, it is known as nucleoid, whereas in a eukaryotic cell, it is known as true nucleus. Okay, it is known as true nucleus. It is known as true nucleus. Okay, true nucleus. It's a true nucleus. Then cell type. Prokaryotic organisms are normally and always unicellular in nature. What is unicellular? They are made up of single cell. For example, bacteria. Okay. And eukaryotic cells with the nucleus can be seen either unicellular or multicellular. They are of both type. Okay. For example, protista, animals, plants, fungus. Okay. These are the examples of the eukaryotic organisms with nucleus. Then their size. Normally, prokaryotic cells are small in the size. Normally microscopic. We have to use microscope in order to see them. But in eukaryotic cell, their size is vary. Okay, their size varies. Their size may vary according to the cell. Maybe small to big. Okay. Cell wall. In a prokaryotic cell, cell wall is made up of peptidoglycan and many other materials. But we are talking about bacteria. Okay, so it is made up of peptidoglycan. And in eukaryotic organisms, cell wall here. Cell wall may contain cellulose or it may made up of chitin. And cell wall is present into plant, into fungus, but it is absent into animal cell. Okay, animal cell do not have, they are devoid of cell wall, and animal cell is eukaryotic organism, and plant cell and fungus cells are also eukaryotic organism, but they have cell wall present in them. Then complexity is simple of a prokaryotic organisms. They are very simple organisms. Okay, but eukaryotes are complex in the nature, and organelles are absent. Okay, cell organelles are absent into prokaryotic organisms, whereas cell organelles are present into eukaryotic organism. Okay, yes. Now let's move ahead. You have to, okay, write and make notes in between. Remember that, okay? Do not just see the video. Otherwise, you might forget it. Now, this is the cell wall. Okay, next structure, cell wall. We have to learn next structure is cell wall. The cell wall is very outer structure of the plant cell. Now, let's directly read this. Cell wall is present in plant cell, bacterial cell, fungal cell, and the diatoms. These are the examples of the cells, organisms in which cell wall is present. Okay. In a plant cell, the cell wall is made up of cellulose. In fungi cell, the cell wall is made up of chitin. See the material, it is made up of chitin. Whereas in bacteria, the cell wall is made up of peptidoglycan. And in the diatoms, it is made up of biogenic silica, the material. Yes. Let me clear the drawing. Now, what do you see? This structure. It is the rigid outer covering of the plant cell. Okay, it is the rigid outer covering of the plant cell, which is called cell wall. It's a rigid structure. It is inner to the cell wall, plasma membrane is present. Means inside the plant cell wall, plasma membrane is present. Outside cell wall, inside membrane, plasma membrane. The plant cell wall is mainly composed of cellulose, which is the complex substance. So plant cell wall is made up of cellulose. What is the function of the cell wall? It provides structural support, strength to the plant. It will protect the plant cell, like we just saw in the plasmolysis video, okay? It is not letting the cell shrink. It is providing mechanical support. Same example, plasmolysis. It will give shape of a cell, okay? It will define the cell shape. Control expansions or shrinkage due to osmosis. Okay, it will not let it over shrunk and over expands. It will help in prevention of water loss. Okay, plant cell wall will not allow the water loss. Obviously, they will be losing the water, but not in high amount. It helps in transportations of the material across the cell through the plasmodesmata. You can see the arrow is showing a small pore-like structure into the cell wall. That pore is known as plasmodesmata. It is allowing two cell structure of a plant cell to contact with each other, through which some material can be exchanged between two cells. It's called plasmodesmata. Cell wall will make barrier between cell internal structure and outer external environment. Okay. When the living plant cell loses the water through exosmosis, there is a shrinkage or contractions of the content of the cell away from the cell wall, which is called as plasmolysis. We can we can perform this activity, and actually we have performed this activity already. So cell wall structure is over along with the function. Now let's move to the next point, which is cytoplasm. Okay, what is this? Cytoplasm. The cytoplasm is a jelly-like substance present within the cell surrounding. Okay, it is present within the cell surrounding by the plasma membrane. So this structure, if you see, okay, this structure between the nucleus and plasma membrane, this yellow looking structure, looking like a watery jelly-like structure, which is plasma cytoplasm. Okay, cytoplasm. Now, it is a medium for the cell metabolism. The cytoplasm is a medium. It is behaving as a medium for the metabolism chemical reactions. Then it consists of water, food, gases, waste material, minerals, some enzymes, etc. It also a place for the cell organelles to function. It's a medium basically. So in that medium, all the organelles are present, and these organelles are known as functional unit of the cell. Let me remind you, these organelles are doing function for the cell. Okay, this is the division of labor. The cytoplasm is colorless structure. In the figure you can see it is looking like a glass. Okay. Okay, see, this is the nice animation view. Maybe you are able to see that. Okay. So everywhere this glass-like a structure, jelly-like structure is cytoplasm. Okay, so cytoplasm is over. Now, let me remind you what we did up till now. We learned plasma membrane, we learned nucleus, and we learned cell wall and cytoplasm. The basic structure of the cell is over. The next part is cell organelles. Cell cytoplasm contains small compartment are called cell organelles. So what are the cell organelles? They are small compartment of the cell cytoplasm. You can, okay, they are the small compartment of the cell cytoplasm. Okay. Now, next we have to move towards the next topic, that is cell organelle. Okay. Cell organelle are the small compartment present within the cell cytoplasm. Okay, you can see this all small components, like you can see this mitochondria, then there is a nucleus, then this endoplasmic reticulum, and this is Golgi body. Okay, so these are the small components present within the cell cytoplasm are known as cell organelles. Let's read. Cell cytoplasm contains small compartment. Okay, what are these? These are small compartment which are known as cell organelles. Okay, these small compartments are responsible for cell functions. What they do? They do the cell functions. Okay, the cell functions are divided by these organelles, and so cell is functional, and a functional cell can make tissue organ functioning, which is known as division of labor. Okay, these organelles are going to do the work on behalf of the cell, and that is how a cell is living, and so we are living. I hope that in a short way this is clear for you. Okay, in this you can also see this cell cytoplasm, and these are the cell organelles. Okay, different cell organelles in different way. Let's move. Large and complex cells include both type of cell, unicellular and multicellular organisms need lot of chemical activity, collectively known as metabolism, to keep their complicated structure functioning. If you do not understand this, okay, I will again explain it. What happens that large and complex cells means both type of cells, either it is a unicellular organism or multicellular organism, eukaryotic organism or prokaryotic organism. They all have to live and function, and for the function they are all doing metabolism. But what happens that these functions are done by separately, differently in both the type of cells. Like in a eukaryotic cell, these all functions are, or metabolism is done by single cell. But whereas the multicellular organism or eukaryotic organism, these functions are, okay, divided among the cell organelles, which is called division of labor. And they are actually doing the function. And to keep these chemical activities separate from each other, these cells uses membrane bound little structures called cell organelle. Now try to understand this. What happens? All the cells will do the metabolism for living, means chemical reactions. But what if all these chemical reactions are collectively occurs together? It will mix with each other. And because of that, there is no result at all. And so these all activities have to be separated from each other. And that is where cell organelles come, okay, into the action, because all the cell organelles are bounded by, or maybe not bounded by, but they are separated from each other from the cytoplasm. And inside them, within them, these activities are happening. Okay, they are doing this chemical reaction separately. And these chemical reactions will now not be affected by the cytoplasm, or cytoplasm will not be affected by this inner side chemical reactions. And these structures are what? Obviously cell organelles. Now there are two type of cell organelles are present: membrane bound cell organelles and non-membrane bound cell organelles. Membrane bound organelle means they are made up of membrane, and non-membrane bound cell organelles means they are not made up of organelles. Sorry, they are not made up of cell membrane, plasma membrane. Okay, there is no plasma membrane in their structure basically. So examples of the membrane bound organelles are endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, plastid, vacuoles. Okay, these are the membrane bound organelles. That means they are made up of plasma membrane. Okay, they are made up of plasma membrane. Just a second. Yeah. And non-membrane bound organelles are ribosomes and centrioles. They are not made up of membrane. So they are made up of what? Protein. For example, they are made up of protein. The ribosome is made up of protein and RNA, whereas centriole is made up of proteins, whereas these membrane bound organelles are made up of plasma membrane. Okay, means membrane is present, membrane is absent. Okay, now remember the names of the both type of yes. So the first organelle, which is a membrane bound cell organelle, is known as endoplasmic reticulum. Okay, it is known as endoplasmic reticulum. Let me put this screen here. Now, what is endoplasmic reticulum? It is the large bound network of membrane bound tubes and sheets as it's shown in the diagram. Let me, okay, because everything is in front of us, so it is easy for us to learn. Again, we have to read and understand. It is what? It's a large, because it is very big. You can see this whole structure, this whole membrane bound structure. It is very large. Okay, it's a membrane bound means made up of membrane. It is membrane. Okay, what you see this line, if I draw for you, okay, let me draw it for you. So if I draw this line where what I'm drawing is what? It is the plasma membrane, means they are made up of plasma membrane. Okay, so it is looking like tubes and the sheets. Okay, it is looking like tubes and the sheets, like in this diagram. This is sheet like structure. Sometimes this is the tube like structure. Okay, you see, this is tube like structure, present as the long tubes, round or oblong bags called vesicles. Endoplasmic reticulum ER is similar in the structures to the plasma membrane. Their structure is same like plasma membrane because they are membrane bound structures. There are two type of the ER, endoplasmic reticulum. Number one is called RER, rough endoplasmic reticulum. It is named RER rough because of the presence of ribosomes on its surface. Okay, so many ribosomes are present on its surface, and so it looks rough under the microscope. You can see in this image, you can see this pink structure. Okay, this pink structure I will show you. These pink structures are ribosomes. You see, because of the presence of ribosomes, it looks rough, and so the name is given rough endoplasmic reticulum. Now the function of rough endoplasmic reticulum is it will synthesize, make protein and hormones, and these proteins then sent to the various parts of the plant as per need of a cell. So basically it will do the synthesis It will make something for the cell, and that is protein and hormones. Second type of the endoplasmic reticulum is SER, smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum. Now it is known as a smooth because there is no ribosome present. Ribosomes are absent. Ribosomes are not present on them. Okay, and because of that, it is looking smooth, not rough. And their function is they will synthesize the lipid, steroid, and hormones. Lipid will help building the plasma membrane because plasma membrane is made up of lipid, phospholipid. And so it is known as membrane biogenesis, means formation of a membrane. Okay. If you don't understand, I will tell you again. Yes. The function is to make the lipid, all the types of lipid like steroids and hormones. And these lipids will help to form the plasma membrane, to make the plasma membrane. And because it is making the plasma membrane and it is possible with the SER, okay, this process is known as membrane biogenesis, means formation of the membrane. Okay. Second membrane bound cell organelle is Golgi apparatus. Okay, Golgi apparatus. It was first discovered by Golgi, Camillo Golgi in 1898. Okay, yes. Don't disturb right now, please. Okay, do not comment right now. Okay, so yes, again, see, I'm saying Golgi apparatus. It is discovered by Camillo Golgi. It is a name of a scientist. Okay, Camillo Golgi in 1898. He said that it is a densely stained structure. He could see it's a densely stained structure. And after him, the name is given Golgi apparatus. Okay. It is always near nucleus. There are few topics we have to underline. Okay, underline. Discovered by Camillo Golgi, and it is present near the nucleus. Okay, it is present near nucleuses. These were later named after the scientist as Golgi bodies or Golgi apparatus. This is Golgi body. You see, this is Golgi body in this image. This, let me give you this. Okay. It's a Golgi. It is looking something like this. And in this Golgi body, you can see this round circular structure. These are known as vesicle. Okay, we are going to draw this. But first, let's see the location and the function. So, location is present near the nucleus and endoplasmic reticulum. Structure of the Golgi bodies, they may consist of many flat or disc-shaped sac-like structure. Okay, they are made up of disc or sac-like structure. Let us draw it. Okay, see, if you want to learn how to draw, this is the way to draw. This is the one sac. What is this? It's a one sac. Okay, disc-shaped structure, maybe flat structure. From the center, it will be something like this, and from the end, it is bulb structure. Now, such many structures are present together on one above another. Okay, this is known as Golgi body. And in the end, you can see there are some round shapes are present, which are called vesicles. Yes, which are called vesicle. Sacs are also called as cisternae. Okay, these sacs are known as cisternae or cistern. Golgi body consists of two 7 cisternae. Now you can see I have drawn 3 cistern. Now such 2 cisterns, okay, or maximum 7 cisterns can be present in the Golgi body structure. 2 means young one, and 7 means old aged Golgi body. Function of the Golgi body is to store, modify, pack the products from the endoplasmic reticulum. Okay, what is made by endoplasmic reticulum? Endoplasmic reticulum will make sugar. No, endoplasmic reticulum will make proteins, okay, hormones and lipid. So these things are modified, packed and stored by Golgi body. Okay, it modifies the simple sugar into complex sugar. It also modify the protein into glycoprotein and lipid into glycolipid. And similarly, sugar to yes. Yes. Okay. So basically these two things, protein to glycoprotein and lipid to glycolipid. Actually, it is also adding some sugar to the protein. Okay. Which is known as glycoprotein. Okay. Why we call it glyco? Glyco means glucose and protein means protein. So when we add glucose into the protein, it is glycoprotein. When we add glucose to the lipid, it is called glycolipid. So you can see sugar is added to the structure. So it is a modification of the both things. And Golgi body forms lysosome. Okay, lysosome is made by Golgi body. Let me clear the drawing. This is the next structure called lysosome. So what is lysosome? Lysosome is again a membrane bound structure. It's a membrane bound structure because it is made up of membrane. So lysosomes are membrane bound structure secreted or released from the Golgi body because Golgi body is maker of them. Lysosomes are waste disposal system of the cell. What they do in a cell? They help in waste disposal. Okay. Waste means whatever the things which are not needed by the cell, it will be removed, digested by the lysosome. Lysosomes are filled by, see, lysosomes are filled by all types of digestive enzymes. Okay, what is there inside? So it is packed by digestive enzymes. Those enzymes are known as hydrolytic categories of enzymes. Okay, for example, protease can digest protein. These enzymes are normally capable of digesting everything. Like lipase is there, which can digest the fat. Then carbohydrates can digest the carbohydrates. And the cell basically consists of fat, protein, and carbohydrate. Now you just imagine the lysosome's function is to digest. It is packed by digestive enzymes like protease, lipase, carbohydrates. Then there are some other enzymes are also present like nuclease is another enzymes. Okay. If by mistake this is broken, okay, inside the cell, and the cell is made up of fat, protein and carbohydrate only. So it is going to digest the whole cell each. Okay. Hence it when breaks into the cell, these enzymes can digest the cell and kills the cell. So it is known as suicidal bag of the cell. Okay. This is the reason we call it suicidal bag. Lysosome also helps to keep the cell clean by killing, digesting any foreign material like old worn out cells, infected cells, injured cells or bacteria in your tissue or body or cells. Okay. So this is the reason we call it disposal system. Okay, this is the reason we call it disposal system. I hope that lysosomes are clear. Okay. Next organelle, membrane-bound organelle is vacuole. Okay. Vacuole is single membrane bound. What is vacuole? It's a single membrane bound structure. Okay. A space found in cytoplasm where we can see that into cytoplasm of the cell. In the vacuole contains what is inside the vacuole? Water, sap, excretory products of the cell and other materials which are not useful for the cell. So inside this vacuole, okay, what is the thing present? It is packed by water, sap, excretory product, food, gas, etc. Some useful material, some non-useful material. Yes. So cell vacuoles are present into various different type of cells like plant cell. So in a plant cells, the vacuoles functions are and how they are. So they are very large in size. Okay, plant cells have large vacuoles. Normally occupy 50 to 90% of the cell volume. Okay, they are very, you can see they are very large from this percentage. They are full of sap. Okay, they are packed by sap and thus provide turgidity and rigidity to the cell. Many important substances are stored in the vacuole. Okay, not only these things, but they store some important substances like amino acid, sugar, various organic acid, and some proteins are also present into vacuoles of the plant cell. Then in an amoeba cell, the vacuoles are known as food vacuoles. Okay, because they contains the food items that amoeba has consumed. Amoeba, when eating the food, it is packing the food into the vacuole, and that vacuole is known as food vacuole present into amoeba. A contractile vacuoles which are important in excretion. Now paramecium, it is also having the same like amoeba. Okay, the vacuoles are same like amoeba. And in animal cell, very small vacuoles are present. Okay, the vacuoles are very, okay. So let's, next is mitochondria. You can see in this picture, mitochondria is present. You can see the arrow. Okay, this is the mitochondria. You can see the mitochondria is having two membrane. One is outer membrane and another is inner membrane. Okay, so the mitochondria is a double membrane bound organelle. It has two membranes, outer membrane and inner membrane. Okay, that is how two membrane. The outer membrane is continuous and porous, while the inner membrane is highly folded into finger-like structure or projections called cristae. And these are responsible for generation of ATP. Hence, mitochondria is known as powerhouse of the cell. Means it's a site of aerobic respiration. The inner compartment means inside the finger-like projections or inside the inner membrane. It is filled with dense homogeneous substance called matrix. The matrix basically we can compare with the cytoplasm of the cell. Similarly, it is the cytoplasm of mitochondria called matrix. The matrix possess circular DNA, means plasmid, a few RNA molecules, and ribosomes 70S ribosomes category. Mitochondria can divide by fission. It is a type of cell division. Means mitochondria can duplicate itself within the cell by itself. Next, we'll be seeing the image. Okay, it is a drawn image. Just a second. This is drawing of diagram. What you see, this red color structure is internal membrane. You can see it is looking like a finger. The fingers are inside. Okay, that is the inner membrane. The inner membrane is surrounded by outside. This black membrane is outside membrane. And what you see, this yellow structure within, it is called matrix. This is this black one. This ring-like structure is small circular DNA, the plasmid. Okay, you can see all the names. This red one is inner membrane. This is crista. Okay, this is a finger-like structure, crista. This is outer membrane. This yellow is matrix. Okay, this is crista, and these black dot-like structures are called ATP synthetase enzyme, which are responsible for ATP formation. Okay, this whole yellow structure is matrix. Next membrane-bound cell organelles is plastid. They are exclusively present into plants only. Okay, plastids are present in plants exclusively, only into plants. They are normally storage granules because they store anything inside them. Okay, so that's why they are called storage granules. They will be storing something, and according to what they store, okay, they are of three types. Means what they are storing. So if, okay, let's directly see the types. One is called chromoplast, other is called chloroplast, and the third is called leucoplast. Now let's see the types here. First, chromoplast. The chromoplast means they store color. Okay, you can see what they store. They store color, and they are present into normally. Okay, they store. Okay, it is a sore. Okay, we have to write T also. Stores colors. They are present into flowers and the fruits because you see the flowers are colorful, fruits are having some colors because of the presence of chromoplast. Chromo means color. Okay, the meaning of chromo is color. So when they store the color, it's called chromoplast basically. Then the second type is chloroplast. Okay, chloroplast present in plant leaf and they store chlorophyll pigment. Okay, where you can see the chloroplast, you might have heard this term previously, okay, in your other standards too. So the chloroplasts are present into plant leaf, and because of them, they are green in the color. Okay, leaves are green because of the chlorophyll pigment. The chlorophyll pigment is responsible for photosynthesis, making of the food, and also it is responsible for green color of the leaf. So this is all about chloroplast. And the third one is called, okay, let's see. Third one is leucoplast. Now basically leuco means colorless. Okay, leucoplast means it is the colorless. They do not store color, and then what they store food. Okay, they store food, and if they store the food, they are present into fruits, seeds, etc. Okay, wherever a food is stored, it is a leucoplast. Now leucoplast again divides into three category: amyloplast, oleoplast, and proteinoplast. Proteinoplast means they store protein. Oleoplast store fat, and amyloplast store carbohydrates. So that is how there are three types of the leucoplast. Next is this green beautiful structure, which is chloroplast. This is the chloroplast. Okay, we just talked about. This is the chloroplast. Let's see the structure first in the chloroplast. This is a coin-like structure. If you see, this is the coin-like structure. This coin-like structure is, okay, this is one coin-like structure. This coin-like structure is known as thylakoid. Okay, it is known as thylakoid. This is the cut section of thylakoid. You can see how it looks. It's look like a coin, and many coins are stacked on each other, and they forming a pile of this thylakoid. So the single is called thylakoid, and when they form piles like this, okay, many on each other, it is called as grana. Okay, this is called as grana, and all the granas are attached with each other like this. You see, they are attached to each other. They are connected with each other. And again, it is also having two membrane. This is outside membrane and this is inside membrane. Two membrane, okay? Outer membrane, inner membrane. And the inner cytoplasm is known as stroma. And you can see that it possesses few structures like the mitochondria, like this is the DNA, okay? This is the DNA extra chromosomal material plasmid. And you can see this small violet dot like structure, which are the ribosomes. Okay, so this is the structure of chloroplast. Now let's see, it is also membrane bound structure because it is made up of membrane. You can see there are two membranes, okay? Outer membrane and inner membrane. So the first line goes double membrane bound structure present into plant exclusively. It possesses chlorophyll pigment. Okay, what is its main function? It will store something. It's a plastid. It will store chlorophyll pigment. And chlorophyll pigment is responsible for the green appearance of the plant, and it's also responsible for photosynthesis. It will help the plant making food. The inner membrane is forming the coin-like structure called thylakoid. Okay, you can see in the diagram, this coin-like structure is formed by inner membrane. And when many thylakoids are forming piles called grana, the stroma, okay, the stroma is jelly-like cytoplasm of the chloroplast, which is in addition contains ribosome, plasmid, and other enzymes which are necessary for photosynthesis. This is chloroplast. Next organelle is non-membrane bound cell organelle. Okay, all the membrane bound organelles are over. Right now, non-membrane bound organelles. That means no membrane present in the structure. One example is ribosome. You can see in this picture ribosome. This violet color big dot like structure is ribosome. So ribosome is non-membrane bound organelle present in both type of cell, eukaryotic cell and prokaryotic cell. Means ribosomes are common to both, but their types are different. Like eukaryotic cell possess ATS ribosomes. Okay, it possess, yes, it possess ATS ribosome. So ATS is in eukaryotic cell, while 70S is in prokaryotic cell. And ribosomes are found freely floating into the cell like in this picture. Okay, you can see this picture. And they are also attached. They are also attached to the endoplasmic reticulum. And this is endoplasmic reticulum. It's a membrane of endoplasmic reticulum. So ribosomes can be free floating into cytoplasm of a cell, or it may be attached to the endoplasmic reticulum, that is forming RER. Okay, it is forming RER. The function of the ribosome is to synthesize the protein. It will make proteins. It's its main core function. You can see in this picture, so many dots are there. Okay, see these dots. Freely floating and attached to the ER. Next structure is here, that is again non-membrane bound cell organelle. The name is centriole. You can see the diagram of centriole in here. Okay, it is looking like a cartwheel structure. You can see this is the centriole structure. You can also see something is coming out, this fiber-like structure from the centriole. So this is centriole, and centrioles are, you can see they are looking perpendicular to each other. Okay, they are two in number and they are perpendicular to each other. Now, centrioles are present into animal cell only. Okay, they are not present into plant, only into animal cell. It is also a non-membrane bound structure made up of protein. It is made up of protein and it is non-membrane bound structure, means made up of protein and not membrane. The function is to help in the cell division. So while in animal cells, when they undergoes the cell division, this is the responsible structure, centriole. Without the centriole, animal cell cannot divide. So this will be all. The chapter is over. Now, if you have any doubt, query, question, you can leave comment. Thanks for watching. Okay, let me give you this.